I don't know if that's working, but we'll see.
Oh.
All right, did you see this?
Oh, yeah, yeah, yeah.
So if they fold up, this goes like this, and then the table goes down.
Oh. Oh. That is.
And then
You want to maybe you should wear some.
what?
I got it. And
I
I
got
will.
I'd rather just not think about things.
Okay then. You know, hold them and I will move them away because I'm not.
Okay. Boy. I don't know, that one's not working. Guess I should have figured that I needed something else. You know what, just put that one off to the side. As good as this is. We will survive.
Hey, no, you want to do this?
Need all these chairs. No, that's not. Right, I'll put the row here. I was gonna put the row down the middle, right there. Actually, let's just take these. I'll put some over here. That look fine?
There are specific faces. Put in front of you.
Actually, Kim is really good during my practice. She had a big smile on her face. So. I think that's good, right? What am I looking for?
Okay, good luck. See you in a.
Thank you. See you in 10 minutes. Hey, Joe. Hello, how are you? Good. What?
Hello.
Hello.
Thanks for having me.
Want to say hi to Kara?
I have to say, I have not been in this room about cookies before, and I'm really disappointed.
Sorry about that. I agree. Is that her?
Oh, P.S. Do you have your form, and did you get it sent to you by your coordinator? Because apparently you can't just print it out from the one online because it's not the right form.
Did not do that. I just have the form that I printed out and wrote in.
Maybe
Is it
biochemistry
the right
thinks
form?
it's the right form.
That's different than my form. Well,
Are you getting a new form signed? Is that right?
Yes.
Who is the biochemistry coordinator now?
We don't know how to say her name.
Stacia.
Stacia, Stacia, Stakia.
I ran into Andrea yesterday. We live in the same neighborhood.
Yeah, that's
I was
fine.
going out
Do you
for a
ever
run.
just-
Oh,
Stacia.
yeah, 'cause
On a
Joe
firewall.
knew she lived over there.
Yeah,
She goes to that Target, she looks all the time.
She was walking to the park that I was gonna go run around with
Nice.
their 2 kids.
Is this gonna be Kara?
Did you do this? He was like, you can say hi to Kara. I was like, that's just your desktop.
Yeah. Wait, is it up though? I mean, I don't know what this is. This is it,
Oh, I didn't know it was actually up.
but I don't see Kara. She might be listening to me though. Who knows?
That would be
I'm
creepy.
sure it's minimized so he doesn't have to stare at her face the whole time.
His own face.
That would be weird. I knew that. Kara wants to me. Sorry, and she's like, it's just like the thing. It's like a lot of things. I was like, I got it. Doesn't say anything. She.
Did you go?
Hello.
You're ready to defend it, but anyway.
I'm A, here to be Joe's friend, and B, thank you, collecting signatures because I printed out the wrong form.
You better hope no one takes it back. Just like copy the other one and take it.
That's what I thought. I was like, can I just like Photoshop the signatures in so I don't have to go find that?
So where's the dissent line again?
No, I didn't have
Oh,
enough
yeah,
people. If you just said
finding.
it doesn't work.
I think Josh Rubin's.
Thank you. Because you have to get it sent to you by your coordinator. They didn't tell us that.
Is Kara on here already?
She said she was.
Oh, well then she's probably, because I pulled up the Hangouts.
Go away.
No, I pulled it up, but
Oh, so like I
My sister.
Googled the form
Oh,
and I
okay.
got all
Hi,
the
Joe's
signatures,
sister.
and my coordinator tried to submit it and they wouldn't accept it. And they,
what?
because it was an outdated form. And they
No,
said that they don't
it
have
doesn't.
the form
I was
on
gonna
the
say
website
either.
anymore because they don't want people to be using the wrong form, so they just send it to you, but they don't tell you when they need to send it to you.
Yeah, I don't
Forward
know.
it to him.
So is this really different
Mhm.
than what you have? We the undersigned. I just don't.
Why don't you just,
I filled
are you
out her
online
entry.
right now? Just email whatever her name is.
And
Peter can do it. Fonts, because.
the words part were the same
Sasha.
on mine.
So what's? Oh, you just need an email.
Peter's gonna email Saskia and get the right one. This is not something that I should know.
Oh, I was like, copy.
And to me, I think too, you like brought a copy to the time. I was like, did you get me coffee?
Do you want me to sit someplace where you can see me? Or?
Depends if we smiling or not.
Depends if you say something funny.
What's that form called again? Thesis defense form?
It's like the examination approval form.
Yeah, examination approval form.
I have 4 to 5 people already. That's not very nice. Because I know she will be here eventually. Is she the one you're missing? Her and Josh were I heard her crunching around and I was like,
Oh,
gosh.
gosh.
And I couldn't find her.
She like disappeared. Maybe she set up like a tape player. Just like sounds of like crunches.
Decoy, she was like DJ Diva situation, so it's
Exactly.
running in the halls.
That's what I would do if I were her.
Search pretty
Always looking.
Looks like there's a new inside.
Stacey called me to be like, oh, you can't take this off of him.
Yeah. Let's see.
Hey,
Good. How are you?
good. My brother was like, oh, great, I'll come. I was like, oh, just come. Yes. Yeah, this is like my invited. Yeah.
Should have texted you. I meant to try to find you an NBA. Yeah, I think we.
I was like, nervous while I was
That's what I guess. No, I guess she actually.
It's like
You know, I'm good, thank you.
Good morning. Welcome, students, faculty, and family. I see a lot of family members here. I think that's great. It is my privilege to introduce Joe Stavella, who is defending his thesis today. Joe's a student in the biochemistry program. And so if he graduates, I think when he graduates, I'll be getting rid of a student out of my program, but will also be graduating one. So it's just a double whammy for me. Joe was born and grew up in Grand Rapids, Michigan. And then he went to college, to Hope College actually, which is in Holland, Michigan. So perhaps it's not a surprise that he wanted to do his PhD with a Dutchman. I'm just taking it because it was an absolute delight to have Joe. At the time he came in the lab, we had a renewed interest in the presence of ribonucleotides in the genome and how they could be removed from the genome because obviously they don't belong there. And Joe decided to look at the largest pool of ribonucleotides in the genome, and those are Okazaki fragments. At the time when he started this, he only had 40,000 foot view of this problem. We were looking down at the forest and it looks green, and we thought that was it. But Joe went down to the forest and to bring this simile back to science, found some remarkable regulatory pathways that he uncovered by really focusing in on all of the steps involved in ligand strand DNA replication. He will present this to you in a 45-minute lecture, and he will make it look easy. But it was far from easy, actually. There were many, many challenges. He will indicate a few of the challenges. And I think what really made Joe into a mature scientist and what I admire so much about him is how he worked through all of these challenges, most of them which he's not going to discuss today. And I remember when we were sitting down in our office and my office, and we were going through one of the problems, things didn't work again. And, we were both shaking our heads, and Joe left. And I was still shaking my heads. And 2 days later, he came back and he found a way that we were going to solve this problem. And this is really the mark of a mature scientist to which he has grown. So he will present this research to you. Then after his presentation, there will be questioning from the general audience. Then we will leave the thesis committee and we'll be questioned a little more, perhaps even grilled. But we will come back here at noon and have a little celebration for Joe. The title of his talk is Timing is Everything. Oh, that's not the title. It is Timing, the Steps of Okazaki Fragment Maturation.
Thank you, Peter, for that really nice introduction and for starting to lay out some of the work that I will tell you. All right, so I don't need to tell many of you in this room, but faithful copying of genetic information in each cell cycle through DNA replication is a fundamental biological process that occurs in all living things. And in each domain of life, cells have evolved complex enzymatic machineries in order to accomplish this goal. And so a long-term aim of Peter's lab has been to understand how DNA replication occurs in eukaryotic cells. So here's a simplified schematic of the eukaryotic replication fork. And in eukaryotes, and in everything I'll be telling you about today is in the budding yeast or Saccharomyces cerevisiae replication system. But the two strands of the DNA double helix are unwound by a very large helicase complex, and this creates the classical bidirectional DNA replication fork. And due to the polarity of the DNA molecule itself, that dictates that one strand of the replication fork is replicated continuously, and this is the leading strand, and the other strand must be replicated discontinuously, and this is the lagging strand. So in budding yeast, synthesis on both strands is initiated by the pol alpha primase complex. And pol alpha with the primase initiates replication on both strands by laying down a short RNA primer that can be extended by the later DNA polymerases. Then on the leading strand, the major polymerase is pol epsilon, and on the lagging strand, the major polymerase is pol delta. And everything I'll be talking about today concerns the lagging strand and how pol delta replicates on the lagging strand. So for most of my talk, I'll be referring to pol delta sort of just as a single entity, but it's actually a very large enzymatic machine with multiple subunits. So I just wanted to go through some of the structure of how pol delta looks like before I go on with the rest of my talk. So this is just a structural model, and it's an assemblage of a number of different structures of different forms of pol delta and also homologous DNA polymerases in this blue domain. But pol delta in budding yeast is 3 subunits. It contains the catalytic subunit, which is pol3, and 2 accessory subunits, pol31 and pol32. So the catalytic core of pol3 contains a polymerase domain, the active site of which is here at the primer terminus, an exonuclease domain, which is important for proofreading for misincorporated nucleotides, and also an N-terminal domain, which sort of has an unknown function but no catalytic activity. Pol3 also has this C-terminal domain, which contains 2 metal ion binding centers. And in particular, this iron-sulfur cluster is required for interactions between pol3 and the next subunit, pol31. Pol31, in turn, binds pol32. And I just want to note that only a fraction of the pol32 structure was resolved in that study. Okay, so pol delta on its own is a low processivity enzyme, meaning that it can only incorporate a small number of nucleotides in any one DNA binding event. And if this held in the cell, then DNA replication would occur very inefficiently because pol delta would be dissociating from DNA very frequently. And so this problem is solved through interactions with another protein, and this is known as pCNA. And pCNA is loaded around double-stranded DNA so that it's encircling the DNA. And when pol delta is bound to pCNA, it's transformed into a high processivity enzyme, and it can replicate hundreds of nucleotides in a single DNA binding event. So in this model, pCNA is sort of in profile, but if we just look at the pCNA ring itself, we can see its ring or donut shape, and this is the central cavity in which double-stranded DNA would pass. Now, another thing to know about pCNA is that it is a homotrimeric protein, so it contains 3 identical subunits outlined in different colors here. So because the interaction between pol delta and pCNA is one of the most important for completing DNA replication, pol delta contains multiple motifs for interaction with pCNA. And I've outlined 2 of those here. So first, the zinc ribbon, which is the second metal ion center in the pol3 C-terminal domain. This zinc ribbon is required for functional interactions between pol delta and pCNA. And also at the extreme C-terminus of pol32, there's a consensus pCNA interacting protein motif. And so these are the 2 most important motifs within pol delta that interact with pCNA. Okay, so just going back to our model of the eukaryotic replication fork, Peter sort of outlined this a little bit, but because the lagging strand is replicated discontinuously, each of the Okazaki fragments, these are the short fragments that make up the lagging strand, but each of these initiate with RNA. And this RNA must be removed and the resulting nick ligated together in order to yield the mature genome. And so pol delta works with additional enzymes that I'll describe in the next slide to remove this RNA, ligate the nick, and complete the process with double-stranded DNA. So how does this happen? So after pol delta lays down a short primer, all of the single-stranded DNA that was exposed by the replicative helicase is bound by the single-stranded DNA binding protein known as RPA. And then pCNA is loaded onto double-stranded DNA with RFC and ATP. Now RFC is a heteropentameric complex, and it's a AAA plus ATPase, which basically means that it uses the energy of ATP to open the pCNA ring at a monomer-monomer interface and loads it around double-stranded DNA. And once pCNA is loaded, pol delta can associate and extend the primer until it reaches the 5' end of the preceding Okazaki fragment. So then when pol delta runs into the end of the preceding Okazaki fragment, it has the intrinsic ability to displace this strand partially and continue replicating. And this is known as strand displacement. And this, in effect, forms a short flap. So pol delta, as I mentioned, has an exonuclease that's important for proofreading, but this also comes into play in strand displacement. Because once pol delta has performed a small amount of strand displacement, it'll actually cut out those few nucleotides that it has inserted, cutting back to the nick position. And so this repeated strand displacement and exonuclease is a cycle that we call idling. And this idling cycle keeps the polymerase at the nick position, preventing the formation of longer flaps so that these flaps can be processed by the next enzymes in the pathway. And the next enzyme is FEN1, or the flap endonuclease. And so when pol delta makes these short flaps, FEN1 will cut these flaps, and it's hypothesized that the repeated strand displacement synthesis by pol delta and then flap cutting by FEN1 will completely remove all of the RNA that initiated the preceding Okazaki fragment. And because during this process, this nick position is translated all the way through the RNA until it reaches DNA, we also call this process nick translation. And I also want to note here that as well as pol delta, FEN1 also interacts with PCNA, and that's very important for its function. So finally, once these enzymes have created a nick, DNA ligase can seal that nick, yielding completely double-stranded DNA. And there are hundreds of thousands of Okazaki fragments in the budding yeast genome, and so this whole process needs to happen efficiently many, many times in each cell cycle. So one of the reasons that we study how this process occurs in the normal circumstance is because we want to understand how the cell prevents this process from going wrong. And really the main way this whole thing goes wrong is through the formation of long flaps. So pol delta, of course, performs strand displacement synthesis, and sometimes the idling cycle is not sufficient to keep the polymerase near the nick. So pol delta can form a long flap on its own, and this can also be aided by helicases such as PIF1. And once the flaps reach a critical length of about 15 to 20 nucleotides, they can start to be bound by RPA. And binding to RPA actually inhibits the activity of FEN1, so not only have we formed a long flap, but FEN1 can no longer process it. So yeast has actually evolved a mechanism in order to deal with this, and this involves the nuclease DNA2, but in situations where this nuclease is not around, long flaps can lead to insertions between direct repeats, and also if no processing enzymes are around, it can lead to collapse of the replication fork and inability to complete DNA replication. So let's go back to this slide. And so as Peter mentioned, as I sort of came onto this project, we had known all of these basic steps, and this has been the work of Peter's lab and many other labs over a number of years. And I was trying to figure out a way that I could sort of go into more detail understanding this mechanism. But I was running into this challenge, and I will readily admit that it's largely a good challenge to have. But this is that pol delta working with PCNA replicates very efficiently. So what I'm showing you here is just a primer extension assay where I have a labeled primer. And when I incubate this primer template substrate with pol delta, DNTPs, and PCNA, and these other factors, pol delta will extend the primer to the end of the template, and every band that it's hard to see on this gel, but every band is a single nucleotide product. And what you can see is that at time 0, all of the material is in the primer position, or +0 nucleotides. And then at 5 seconds, where, which is basically the fastest time point I can take by hand, essentially all of the material is in the +50 position. So we're missing a lot of things between 0 and 5 seconds. And so Peter and I sort of talking about this problem together, hypothesized that not only with replication, but with all the steps of Okazaki fragment maturation, we thought we were missing a lot of mechanistic details because we just didn't have the kinetic resolution to look at this process. So in order to kind of alter our traditional in vitro replication techniques into a technique that had a higher kinetic resolution, I turned to rapid quench flow kinetics. And here is a schematic of the rapid chemical quench flow apparatus. And I know that it looks very complicated. We have lots of syringes, lots of lines and loops and stuff. But at the end of the day, what this whole machine really allows you to do is mix two samples very quickly and take very fast time points. And so you put one sample in one side, one sample in the other side, the machine mixes them together and shoots them out into your tube at defined times. And so throughout this talk, I'll be showing you diagrams that look like this, which outline the experiments that I'm going to do and also mirrors the actual setup of the quench flow. So in the most basic reaction, on one side of the instrument, I'll have nucleotides, and in all the experiments I'll show you, I got saturating nucleotide levels. And then on the other side, I'll have my polymerase incubating with DNA and possibly accessory factors. These will get mixed for some time, quenched in my tube. And instead of 5 seconds now, the fastest time point I can take is 5 milliseconds. So we've really increased the kinetic resolution of what we can look at. So as Peter alluded to, this adaptation to the quench flow apparatus was not completely seamless. And there were a few challenges that we needed to overcome, and I just want to highlight a few of them. So as I mentioned, Pol Delta has a polymerase domain as well as an exonuclease domain. And this has a few different functions that I talked about. But when I'm performing a quench flow reaction, I need to incubate pCNA, Pol Delta, and DNA all together in the absence of nucleotides for long times before I start my reactions. And in that situation, the exonuclease of Pol Delta will actually completely chew up my primer. So when I want to start the reaction, I won't have any starting material left. So for that reason, in basically all the experiments I'll be showing you, I'm using an exonuclease deficient mutant of Pol Delta. And this is D520V in the catalytic subunit Pol3. I'll be showing you one experiment with the wild type polymerase, but I'll describe the workarounds we used to do that later. All right, second, pCNA is loaded onto double-stranded DNA by RFC and ATP. But once again, when I'm incubating all of these things together for the long times, ATP can actually be a substrate for Pol Delta. And so I end up with, when I start the reaction, I end up with this ladder of products at time 0. And so I wasn't getting a good starting point for my reactions. And so instead of ATP in these reactions, I'm using this ATP analog, which is known as AMP-CPP. And this analog can still be hydrolyzed here to be used by RFC to load pCNA. But instead of an oxygen here, I have a methylene bridge, and that means that this molecule cannot be incorporated into DNA by Pol Delta. And then finally, when pCNA is loaded onto double-stranded DNA, it can diffuse along it. And if my templates are not topologically constrained, pCNA will slide right off the end of the template. So in each of my templates, I have 5' and 3' bioconstrictavidin linkages on the template strand. And these act as bumpers, keeping pCNA on double-stranded DNA. So just to sort of summarize, this is the basic system that I'm using. I'm using these short oligonucleotide DNA templates where I have a labeled primer, either with a radioactive phosphate or a fluorescent Cy3, so I can monitor primer extension by Pol Delta. And I'm loading pCNA with RFC and AMP-CPP, and I'm coding the single-stranded regions of my templates with RDA. All right, so first, I'll look at DNA synthesis by Pol Delta pCNA. So the first question that I wanted to ask was whether being bound to pCNA actually alter the rate of the intrinsic nucleotide incorporation of pol delta. And the only way to do this is to look at a single nucleotide incorporation because of the vast processivity differences between the two forms of the enzyme. So I designed this template so that if I just give the polymerase DTTP, it will only incorporate a single nucleotide. And then in one side I have the nucleotide, on the other side I have this mixture of the DNA, pCNA, and pol delta. And here are the results. You can see that at time 0, all of the material is in the primer band, and then you can see this nucleotide incorporated over time. And if you quantitate this data, and now I'm plotting the +1 product over time, what I hope you can see is that when bound to pCNA, pol delta incorporates this nucleotide much faster than pol delta alone. And in fact, this rate is, the best we can get is greater than 300 because this rate is even faster than can be resolved by the French flow apparatus. And so this is interesting because it's really the first indication that pCNA may not simply localize proteins to DNA and stabilize them on DNA, but may actually alter the catalytic activity of a client enzyme. So next, I wanted to move on to looking at pol delta replicating a longer stretch of DNA. So on this same exact template, it's designed in a way that if I add DT and DA, pol delta will replicate 21 nucleotides. And once again, I'm preforming this complex. And so you can see the data for this complete reaction here, pol delta extending the primer over time until it reaches the +50 or the 50 position. And in order to represent this sort of data more quantitatively, what I'm showing on the right is the median extension. And this data corresponds to the black curve of the complete reaction. And what I'm plotting there is really the median of all the products that pol delta makes. And this takes into account that each of these pol delta pCNA complexes are not moving in perfect synchrony. However, if I plot the data in this way and fit to a single exponential, I obtain a rate of approximately 340 nucleotides per second at saturating nucleotide levels. So pol delta can replicate extremely efficiently. So next, what I wanted to look at is whether the single-stranded DNA binding protein RPA either stimulated or inhibited the rate of pol delta pCNA. And I just want to note that this is a homopolymeric stretch here, and we do know that RPA does aid in melting out secondary structures from single-stranded DNA. We wanted to look at a simpler sequence. And so when I drop RPA out of the system, I don't see a substantial difference in the rate of movement of pol delta pCNA, indicating that RPA does not stimulate nor inhibit synthesis by pol delta pCNA. And just for comparison, I'm showing the reaction with pol delta alone, which of course is incorporating slower, but also has a very low processivity, and so it's incorporating much less efficiently. All right, so now I'll move on to the first step of Okazaki fragment maturation, and this is strand displacement. And so, as I told you before, strand displacement in the wild type scenario, strand displacement is balanced by the exonucleolytic cleavage, and this is idling. But I'm using this exonuclease deficient polymerase, and so at least in these first experiments, I'm only looking at forward strand displacement rate. So in order to do this experiment, I modified my DNA templates just a little bit. I once again have a labeled primer, but now I have a blocking oligo here that initiates with 8 bases of RNA. And this mimics the 5' end of an Okazaki fragment. And then in this case, there's a short 2 nucleotide gap between the end of the primer and the beginning of the block. And so here's the data here. You can see that pol delta rapidly replicates through the gap. And this is the Nick position where no strand displacement has taken place. And everything above it are strand displacement intermediates. So pol delta is forming a flat. And so to represent this more quantitatively, I took the fractional occupancy of each replication intermediate and plotted that over time. And what I'm showing you is just the fractional occupancy of select strand displacement intermediates. So what I'm showing here is the Nick position, the +1, the +2, and then the +3 and greater strand displacement intermediates. And these are plotted on a log scale. I just want to point out. So you can see qualitatively and then also more quantitatively when we fit these curves in a model-free way is that first thing that we noticed is that pole delta stalls significantly at the Nick position before it initiates strand displacement synthesis. And so this isn't something that we really had any idea about before. Some people had proposed that pole delta may seamlessly initiate strand displacement, but this does not seem to be the case because pole delta significantly stalls at the Nick before any strand displacement intermediates are made. But then what we observe when pole delta does start to do strand displacement synthesis is that with each nucleotide that's displaced, the rate of forward movement of the polymerase slows 5 to 10-fold with each step. So actually, the growing flap is inhibiting the forward movement of the polymerase. So not only does idling by the wild-type polymerase keep the polymerase at the Nick position, but this, this growing flap, which acts as a brake on the polymerase, is also preventing the formation of longer flaps. All right, so the next thing that I wanted to look at is what structural determinants of the blocking sequence influence the strand displacement rate. And the two things that I wanted to look at were the sugar identity, or whether the 5' end of this block was RNA or DNA, and also the stability of the duplex. So as anyone that's done PCR knows, depending on the sequence, the stability of the duplex that you form will be different. And so I designed 4 templates in order to try to tease out these 2 variables. And these templates had 2 sequences, so one in red and one in blue. And these differ only in the 4 bases at the 5' end of the block. And each of these either have 8 bases of RNA and then DNA or are completely DNA. And so interestingly, people have calculated the dinucleotide stabilities of both DNA-DNA duplexes and then RNA-DNA hybrid duplexes. And depending on the sequence, in some situations, the hybrid duplex will be more stable. And in other situations, the normal DNA-DNA duplex. And so I have each. In this RNA DNA duplex is actually stable and select time points here. And then what I'm showing here is just the advanced strand displacement intermediates. So you can see some differences qualitatively between the templates. And this is the fraction of +4 product and more. And so what I see is that there does not seem to be any obvious dependence on the sugar identity. And so some people had hypothesized that because pole delta is involved in Okazaki fragment maturation, it may have evolved the ability to displace RNA from DNA more readily than DNA from DNA. But this doesn't seem to be the case, because what we see is that when I flip the stabilities of the duplexes at the 5' end, I flip which template is actually displaced faster. And I know that these are just 2 sequences, but we've looked at other sequence contexts as well. And when we compare RNA and DNA, we always see that the less stable duplex is displaced more readily, and it doesn't go along with the sugar identity. All right, so of course, the normal polymerase has an active exonuclease, and I wanted to do at least one strand displacement reaction with the wild-type polymerase to look at idling. And so, as I mentioned before, the wild-type polymerase will chew up my primer, and then since I have a block, the polymerase will chew up my block as well. But at the extreme 3' end, of the block and the primer, I have a C and a G. And so in order to use the wild-type polymerase, I'm forming my complex of DNA, pCNA, and full delta as before, but I'm also adding a small amount of DC and DG so that full delta will idle at this position and this position and not completely chew up my, my template. And then I start the reactions with high levels of all four DNTPs. All right. So in order to look at this, I compared the case of when full delta had an inactive exonuclease to when it had an active exonuclease. So this is data similar to what I showed you before of the exonuclease deficient full delta. What you can see is that after replicating through the gap, in this case it's 5 nucleotides, the nick position accumulates, but this goes down as strand displacement occurs. So then you see the +1 product coming up and down, and then the +2 and greater just accumulates over time. And this, of course, is different in the case of the wild-type polymerase. So what we observe is that, of course, the nick position comes up, but then we see a stabilization of the nick position as these strand displacement intermediates are made. And this is, of course, due to the fact that when the polymerase starts to initiate strand displacement synthesis, it will start to reverse and then stabilize this nick position product. So really, this equilibrium distribution of products is stable through most of the time course. And of course, we knew that this occurred, but we really didn't appreciate how quickly this equilibrium forms, really within 200 to 400 milliseconds, and then it remains stable pretty much through the rest of the time course. And one other thing that we can see from this experiment is that the fraction of nick position is roughly equivalent to all of the strand displacement intermediates. And that means that, at least on this sequence, the rate of forward movement of the polymerase is roughly equal to the backward movement, which once again retains the polymerase near the nick position, preventing long flat formation. But with what I told you before, that the growing flap actually inhibits the forward movement of the polymerase, this actually turns out to be crucial even in the case of the wild-type polymerase, because if the polymerase moved forward with this rate at every position, then the polymerase would form a much larger distribution of strand displacement intermediates and flat products that could increase the propensity to form long flaps. So really you need these two mechanisms working together to keep the polymerase close to the nick position. So next we'll add in another enzyme to the process, and we'll start to look at nick translation involving full delta and the activity of FEN1. And so the first question that I wanted to ask in the context of nick translation is this: what is the FEN1 substrate in nick translation? So I'm going to walk you through this. Most people, when they draw how nick translation occurs, they draw full delta making this flap. In this case, I'm showing a 1 or a 2 nucleotide flap, and then they show FEN1 cutting this flap. But many people through structural studies have showed that this is not actually how things occur. FEN1 actually cuts substrates with a 3' extra helical nucleotide. And then if there's only one nucleotide displaced, it will cut one nucleotide into the duplex. And if there's more than one nucleotide displaced, it will extrude one 3' nucleotide, and then it will have a variable length second 5' flap, still cutting at the same position. And so what many people have showed is that FEN1 definitely prefers these double flap structures. So these sorts of structures are cut more avidly than just a single 3' nucleotide with FEN1 cutting into the duplex. However, what previous work from Peter's lab had showed is that the major product liberated by FEN1 was a mononucleotide, which likely came from just a single nucleotide displaced. And so there was sort of an apparent dichotomy between these different results. And so I wanted to ask, in my system, what is FEN1 actually cutting? So in order to do this, I'm forming this complex as before on my strand displacement DNA templates with BCNA and pol delta. And then in the other side of the instrument, I have nucleotides, and then I add Fen1 at time 0. And here is the data. Fen1 replicates through the gap, forming short flaps, and then the action of both pol delta and Fen1 allow extension of the primer all the way to the end of the template, a little bit at 2 seconds. So in order to address this question of what Fen1 is cutting, I'm once again going to compare the case where I don't have Fen1 to the case where I do have Fen1 in my experiment. And so here are just 3 strand displacement intermediates in the case of just strand displacement, so there's no Fen1. So once again, the Nick position accumulates and then slowly comes down as strand displacement initiates on this template. Now, because Fen1 requires the flap substrate in order to act, we would expect the addition of Fen1 to not alter the fractional occupancy of the Nick position at all. And that's indeed what I see. So this Nick position curve is basically superimposable. So we'll get rid of it. Now, if Fen1 were able to cut a product that came from just a single nucleotide displacement event, we would expect this curve to shift down. The formation should be largely unaffected, but then the decay should go quicker because this flap is being processed by Fen1. And that's indeed what I see. So this provides evidence that Fen1 is actually cutting a flap that comes from just a single nucleotide displaced, even though that it's not its preferred substrate. And of course, we also see a quicker decay of the longer strand displacement intermediates. And providing further support for this, I labeled the 5' end of the block. And this allows me to look at the products that are liberated by Fen1 at the end of the block. And what I'm showing here is the 1 nucleotide 5' product. What you can see is that this product is produced with kinetics that come after the formation of the 1 nucleotide flap, as we would expect, but before the formation of the +2 flap, providing further evidence that this product emanates from just a single nucleotide displaced. So as I said before, the reigning model of Nick translation is that it requires the concerted action of both pol delta and Fen1, this repeated cycle, as it translates the Nick all the way through the RNA or all the way through the template in this case. And in order to look at that in a little bit more detail, what I did is, in addition to labeling the 5' end of the primer and looking at primer extension in the presence of Fen1, this is the data I showed on the previous slide, I also labeled the 3' end of the blocking sequence. So rather than labeling the 5' end, which only allows you to look at the product liberated at the very beginning of the block, this allows one to look at Fen1 chewing up this block. And if Fen1 were acting repeatedly throughout the displacement of this blocking strand, we would expect a ladder of products to form on this strand at longer times. And this is indeed what I observed. So you can see over time, Fen1 is increasingly cutting this substrate, and we end up with a ladder of products at the longest times. And in order to show the tight coupling of pol delta action to Fen1 cleavage, what I did is I calculated the median primer extension past the Nick position and also the median length of this blocking oligo. And that's shown here. And what you can see is that if we fit this just to a simple line, the forward movement of the polymerase is roughly equal to the cutting of Fen1, once again indicating the tight coupling of primer extension by pol delta and to cleavage by Fen1. So all this brings us to this model, which is that multiple mechanisms are working together to limit long flap formation. So as I told you, after pol delta performs the majority of DNA synthesis of an Okazaki fragment, it will displace this first nucleotide fast relative to later strand displacement events. And so this mechanism, in combination with the pol delta exonuclease, keeps the polymerase close to the nick position, waiting for FEN1. And then I told you that the major product for FEN1 results from just a single nucleotide displaced. And this likely needs to re-equilibrate from a single nucleotide 5' flat to a single nucleotide 3' flat. We don't know now whether this is an active or passive process. And what we would predict from other studies is that FEN1 actually cuts this sort of substrate slower, but if pol delta does make a longer flap, FEN1 should cut these double flap structures more avidly, once again further preventing the formation of long flaps. All right, for my final piece of data, I want to switch gears just a little bit and take us back to the structure of PCNA. So as I mentioned before, PCNA is a homotrimeric protein, so it contains 3 identical subunits. And PCNA not only interacts with pol delta and FEN1, but dozens of other DNA metabolism enzymes. And so because PCNA has so many different client proteins, people have proposed that perhaps more than one protein can bind to PCNA simultaneously. And this would increase the efficiency of processes that involve more than one enzyme. And perhaps the best studied PCNA-dependent process that involves more than one enzyme is Okazaki fragment maturation. And so this model is sometimes called the tool belt, and where PCNA would be coordinating different enzyme tools simultaneously. And this tool belt model, in the case of Nick translation, would predict that pol delta and FEN1 can simultaneously bind to PCNA, and then they would alternate access for these DNA intermediates and leading to more efficient Nick translation. However, the opposing model is the sequential binding model. And in this model, the enzymes need to perhaps only bind PCNA one at a time. And so they would alternate access for PCNA, perhaps due to steric concerns. So in order to look at this directly, I wanted to perform an experiment that would try to see if FEN1 could remain stably bound to PCNA throughout multiple rounds of Nick translation. So in order to do that, I preformed a complex of DNA, PCNA, pol delta, and FEN1. And then upon initiation of replication with nucleotides, I challenged FEN1 with an oligonucleotide DNA trap in excess. And this trap had the structure of FEN1's preferred substrate. And then if during the course of replication or Nick translation, FEN1 dissociated from PCNA, it would be bound to the trap and would not be able to act further on these substrates. So this is indeed what I did, along with all of the necessary controls. So I'll walk you through this flowchart here. As I said, I'm forming a complex of DNA, PCNA, and pol delta. When I start this complex with nucleotides, these are the strand displacement intermediates that I observe at 5 seconds. When I challenge this complex with this FEN1 trap, what you can see is I get the same strand displacement intermediates, indicating that the trap does not act on pol delta bound to PCNA. When I pre-incubate FEN1 with the trap, and then add that along with nucleotides, I don't get any products that are longer than the strand displacement, indicating that this trap is effective for FEN1. When I add FEN1 to this complex 5 seconds before initiation of the reaction with DNTPs, I observe extension of the primer all the way to the end of the template, so this is indicative of Nick translation. So finally, when I add FEN1 and I challenge it with the trap, I obtain products that are longer than the strand displacement intermediates alone, and all these products here are indicative of FEN1 processively bound to PCNA through multiple rounds of Nick translation. And I will readily admit that this processivity is not complete because there is a significant amount of the product that stays sort of at the strand displacement position. But what I want to note is that Fen1 must bind to PCNA when pol delta is sitting here. And so what these products really indicate is that Fen1, in some situations at least, can remain bound to PCNA as pol delta replicates through the gap, forms a flap, and then working together, they can perform recessive NIC translation. All right, so finally, I just want to summarize everything that I've told you, and I hope that I've been able to convince you that using these rapid kinetic techniques has allowed a greater insight into how pol delta and other enzymes replicate on the lagging strand. So first, I talked about pol delta replicating with PCNA, and how PCNA may not just localize pol delta to DNA intermediates, but may actually stimulate the catalytic rate of the polymerase. And it's likely that in the terms of all of lagging strand replication, the actual synthesis of the Okazaki fragment is not rate limiting, because pol delta PCNA will replicate this stretch of DNA, which is estimated to be about 150 to 200 nucleotides relatively quickly. And then I talked about strand displacement synthesis, where two mechanisms work to keep pol delta near the NIC position. Not only the exonuclease, but actually the fact that the growing flap inhibits the forward movement of the polymerase. And then finally, I talked about Fen1, where I showed evidence that the major substrate for Fen1 emanates from just a single nucleotide displacement event, but we would hypothesize that when pol delta makes longer flaps, Fen1 should cut these more rapidly, although we haven't been able to show that explicitly. And then also, I showed evidence that even though everyone always draws pol delta and Fen1 this way simultaneously bound to PCNA, that in some circumstances, this may actually occur. And all of these mechanisms together work to perform efficient ligation of the resulting NIC, yielding the mature genome, and once again, prevent the formation of these potentially dangerous long flaps. So with that, I want to turn to my acknowledgements. And obviously, lots and lots of people to thank. Nobody can go through this process without the help of many, many different people. So obviously, I want to thank the Burgers Lab. I'll talk about them more on the previous slide, but I just want to give a shout out to Andrea, who's here. She was a rotation student with me and worked on some pol delta stuff in her time through our lab. I want to thank my thesis committee in advance for all of their wonderful suggestions throughout all of our meetings and helping me kind of formulate this project. And then you'll see that Roberto is not only on my committee, but he's a collaborator. We've worked together on several different projects, and also none of this work would have happened without him because he basically let me steal his quench flow, and it's like in our lab now, so I'm just really grateful that he was so generous with his equipment. Because you can move it back tomorrow. I can move it back tomorrow. Yeah, I do. So I've also had the opportunity to work with many different labs, not only Roberto's lab, Katrina and Saurabh specifically, but also labs in Israel, in California, and in Germany, and different aspects of pol delta biology. Peter and I had the opportunity to go visit this lab in Israel just a few weeks ago, which was an amazing experience. Also, I'm very lucky that Peter's lab has been well funded in my time throughout it, and I want to specifically call out the Binational Science Foundation, which funded my project specifically. So, more personally, working in Peter's lab has really been just a great time. You can see various iterations of the Burgers Lab over the years and having fun at different meetings, and this is Justin like pretending to eat a critter refund. But I'm just really lucky, Peter, that I've been able to work with you throughout the years. I know it must be incredibly difficult to sort of balance like pushing a student with like when they're extremely frustrated and don't understand where things are going. And I just know that I've been able to achieve things that I really didn't think were possible, sort of, when I didn't even know where this project was going. And I thank you for that. And then Bonnie, Peter's wife, is just always so positive and just brightens up the lab. And Carrie, on her phone right now. I mean, literally not a day goes by when Carrie like knows where something is and I don't, and you'd think I know where everything is at this point. But not only because you know where everything is, but it's been great working with you. And then, of course, my fellow graduate students, Justin in my first part of the time in the lab, and then Saran now, been just great grad student colleagues and talking about data, and yeah, I really appreciate both of their collegiality. And then numbers of different postdocs who passed through the lab, Rachel, Elias, and Tanamoy. I'm sorry, Tanamoy, we need a new picture so that you can be on the slide. I just appreciate your presence in the lab and learning more about where everyone comes from. It's just been a good experience. So then to all of my friends that I've made in St. Louis, it's been really great to meet just a number of different scientists, but not only talk science, but just commiserate and share happy life moments. And I don't know who this stranger without a beard is. Snuck into my picture. I just really thank you guys for your friendship and support. And then finally, of course, friends from all walks of life, mainly that I met in college in Michigan, and also my family, my parents, and my dad's siblings that are here, and my sister who couldn't make it, and this is us in the past. She posted this to Facebook today. I just thank everyone for all of their support, even though they don't really understand what I do at all. There's like, you can do it, and I really appreciate that. And I could probably talk a long time about this, but I can talk about that later. And with that, I'll take any questions.
Thank you. Are the questions from Eric?
So do you think PCNA is actually increasing the catalytic activity of Pol delta, or is it merely increasing the lifetime at the prime or template junction?
So what I sort of glossed over is that I'm using saturating polymerase, so it should be bound. So I do think that it's actually increasing the catalytic rate. I can't really use a trap to look at this because when you use a whole variety of traps with Pol delta alone, you don't get any synthesis in any circumstance. And so it's kind of hard to say whether the trap is active. And so just because when I've looked at things, the binding of the polymerase to the template is saturating, that's why we conclude that there's an actual stimulation.
Did the PCNA3 have threefold symmetry?
Oh, geez.
And if so, I was wondering how to bind. Yeah, so how to bind both at the same time.
So, okay. So the main site of interaction between PIP containing proteins and PCNA is in this region here. And so proteins would bind here, here, and here. Does that make sense?
So good.
Yeah. Beautiful work. I wonder if you added the eating a ligase to your reaction already, and then that if you did, then where at which position the ligation happens?
Yeah, so we didn't add ligase, and there's a reason for that. And that's that in vitro reactions, the yeast DNA ligase seems to not really require PCNA. So when you look in the cell, the ligase does have defects if you delete the PCNA interaction motif, but that might be due to sort of just general localization to the replosome. But if you look in vitro, the position of ligation is very dependent on just the concentration of ligase that you put in. So for that reason, we kind of just left that out of these studies.
The second question is about whether you have any evidence suggesting that the PCNA actually can rotate on the template so that you have a sequential strand displacement and DNA synthesis and flat cutting activities occurring.
Yeah, we haven't looked at that. When people have looked at PCNA diffusion along double-stranded DNA, they have some evidence that the PCNA actually tracks with the helix. So it could be some sort of tracking with the helix. We haven't looked at like rotation around at a stationary position.
When you were showing the fractional occupancy
Yeah.
plus or minus Fen 1, I think I missed something there because you mentioned that the fractional occupancy of the NIC site didn't change, but then the other ones changed.
Yes, exactly.
But they all have to add up to 1.
Yeah.
So are you measuring it, is it really a fractional occupancy or is it a fraction of the starting material?
No. So what I do is, what I'm showing is the NIC +1 and +2. But what would add up to 1 is this entire length. But I'm only showing the select things.
So where does so when you get cleavage with Fen, in the presence of Fen 1, you lose +1 and +2, right? Where does that go?
Oh, it goes to these further ones because then it moves faster. So it goes up faster.
I see.
So that means that the polymerase is able to extend.
I see. And then I just wanted to go back to Eric's question.
Yeah.
Did you say that in the presence of trap for Pol delta, you basically see no synthesis? So doesn't that suggest that Pol delta, even at saturation, is coming on and off?
It could alternatively
Very
suggest
quickly.
that because Pol delta has multiple DNA binding motifs, that all traps are active. So
Sorry, say that again.
Pol delta can bind in the exonuclease, it can bind in the polymerase, and so I don't think that you could say either way.
And what is the difference in processivity? I'm not sure if you mentioned plus or minus PCNA.
Well, of course, that depends on salt concentration, but at the concentrations that I'm using, Pol delta alone will go no more than 10, and Pol delta with PCNA, if you do it on a mixed sequence DNA, you'll get 200 to 600 nucleotides. It's more diffuse distribution, but it's hundreds of nucleotides.
What you're working on is basically the last steps of Pol delta, synthesizing the Okazaki fragment. So at this point, it dissociates from DNA. So is your work in anywhere bearing on that process? Is for example,
So actually, Pol delta does not dissociate from DNA. And I'll show you that. That's here. So what I also, so there have been models that have been proposed in the field that when Pol delta initiates strand displacement synthesis, it will have a weaker affinity for flap intermediates, and it will then dissociate from DNA. And so I wanted to look at this directly, and so I used heparin as a trap for Pol delta bound to PCNA. And what you can see is that, to cut to the chase, minus heparin and plus heparin, Pol delta can perform the same amount of strand displacement synthesis, indicating that it's processively performing strand displacement synthesis. And we don't think that there's an appreciable dissociation of Pol delta upon strand displacement synthesis. So a lot of this collision release stuff has been done in prokaryotic systems, and that might occur there, but at least for this system, we don't think that Pol delta, we don't think that forming a flap induces dissociation of Pol delta.
so and that is there a hypothesis how it how it leaves this site? It doesn't stay there, it has to start the next one.
Probably after ligation.
After ligation is what you mean, right?
Perhaps then they would be weaker.
Any more questions? Well, let's thank you. And please come back here around for celebration.
See you later.
We're gonna go down to.
Yeah.
Well done.
It's